Okay, thanks very much. Hi, this is the Cancer Patient Lab. I'm Roger, and today we have a guest to talk about navigating pancreatic cancer. Before we get started, I'll just mention that this is all just informational. It's not medical advice. Um, and also, we are recording this and it'll be posted publicly. So keep that in mind when you stop your video or put your name on your screen. And then finally, Cancer Patient Lab is a nonprofit organization, so we would encourage you all to donate so that we can keep doing programs like this. So, as I said, our guest today is Dr. John Strickler. He is an associate professor of medicine in the Division of Medical Oncology at Duke University and the co-leader of the Molecular Tumor Board and Precision Cancer Medicine and Investigational Therapeutics Research Program. So he is very qualified to discuss the recent advances in pancreatic cancer. So John, with that, and our format generally is we have a little bit of presentation and then we have some Q&A after the presentation. So save your questions, put those in the Q&A box or ask them live after John's finished speaking. So Dr. Strickler, I'll let you take it away.
All right, thank you so much. Pleasure to be here today. And let me just go ahead and share one of my slides here. And I think there are a lot of resources for more basic topics around pancreas cancer and epidemiology, prevention, treatment, those kinds of things. And what I wanted to do is bring my own personal spin on pancreas cancer, which is around what I consider to be the future of pancreas cancer, and that is our era of precision cancer medicine now coming to pancreas cancer, what that means for patients and families and physicians. So before diving too deeply into this precision medicine future for pancreas cancer, it's important to look backwards. I mean, sorry, I have a pager going off here as we speak. When you look backwards at how we treated cancer, certainly before 1950, the treatment for pancreas cancer was really primarily surgical, and that's Osler up there, and he was a turn of the 20th century famous surgeon who basically masterminded these very aggressive surgical techniques to try to remove or cure cancers, and basically was this forerunner of this era of highly aggressive surgeries. But I think we can all see, particularly with pancreas cancer, that that's rarely a curative modality. And around 1950, some new treatments came into play. 1950s, 1960s, chemotherapy, radiation, supportive care got better. And now we're in an area where we're bringing in immunotherapies and targeted therapies. And for a disease like pancreas cancer, which is so rarely cured with surgery alone or with chemotherapy and surgery alone, it's important to know that this future that we're looking at is probably going to be the way that we can really move the needle on a disease that's largely been considered to be incurable for most people before, up until this point. And so what do we mean when we say personalized cancer medicine? And to boil it down, this is the idea that you can bring the right treatment to the right patient at the right time, potentially sparing people toxic therapies that are ineffective and giving them hopefully therapies that are geared to an individual tumor mutational profile so that you minimize toxicity and improve activity. So, I think that's really this idea. Now, pancreas cancer has been very slow to the party with personalized cancer medicine. So in 2013, I was a couple of years on faculty at this point. This was our pie of actionable targets for pancreas cancer, basically nothing. There was no reason to do profiling because there was really nothing to target. Fast forward 10 years later to last year, I stopped in 2023 because 2024 is going to be a very different experience. But now we've got some FDA-approved therapies really to target what we call germline BRCA mutations. And so that's been a big breakthrough in the last few years. KRAS G12C therapies, it's very rare, but they are now available to us in the clinic. It's just that it's 1 to 2% of patients. But then we have some other alterations where we have pan-tumor approvals like MSI-high disease for immune therapy. And then in rare cases, we have fusions. And I'll go into those in a little bit more detail. And then as of last year, there was this emerging target category as well, which I might speak about a little bit as well. So why do we need to start checking these tumors for driver mutations? Well, increasingly it's becoming clear that when you sequence a tumor, look for mutations in a cancer, even a tough to treat cancer like pancreas cancer, when you do the profiling and you find something actionable, you can improve survival. So this was a multi-year analysis by a friend of mine, Dr. Mike Fishbane at Johns Hopkins. He profiled several hundred patients, and when there was no driver mutation identified, the survival was not great at 1.3 years. When there was a driver that was present but not treated, survival was about the same, so no impact on survival. But when you profiled a cancer and treated that with target therapy, survival doubled. And that's with a better quality of life as well, because oftentimes these targeted therapies are much better tolerated than chemotherapy, which is the current standard of care for pancreas cancer. So this is where we were, and this is where we're headed. We are trying to improve survival with sequencing these cancers, finding drivers, and then targeting those drivers with breakthrough drugs. So I had mentioned the germline alterations, but I think what we're seeing now in 2024 is potentially a paradigm shift in precision medicine for pancreas cancer, and that's around targeting something called KRAS. And I'll explain what KRAS is all about, okay? So KRAS is a protein that's present in cancer cells and also throughout normal cells in the body. And I like to think of KRAS, which is shown right here, as a light switch. So a light switch is designed to turn off or turn on, depending on if you need that light bulb on or off. But when the light switch is broken and RAS has a mutation, the light switch is perpetually on. And no matter how hard you flip that switch, it is stuck in the on position. And this has plagued us for decades. Now, 90% of pancreas cancers have a KRAS mutation. So if you were to find one mutation to target in pancreas cancer, this would be it. The problem is that it's taken us literally decades to figure out how to turn off this light switch, because there are so many backup mechanisms. Even if you unscrew that switch from the wall and try to shut it off, there's like backup ways to keep that light turned on, even if you tear the switch out of the wall. So that's really what KRAS is all about. KRAS is a switch that is stuck in the on position, and this is the main driver for pancreas cancer. So when I say that this is the mutational landscape of pancreas cancer, 87% of pancreas cancers have a KRAS mutation. 13% are wild type. I'll talk about these. Wild type means not mutated. I'll talk about that in a moment. And then when you look at the specific types of mutations, I listed these top to bottom: KRAS G12D, V, R. These are the main driver mutations in pancreas cancer. Accounts for about 3/4 of all pancreas cancers. Now down the list, we have KRAS G12C, and here we do have FDA-approved therapies, but look at how often you see them in pancreas cancer, just 1% of patients. So if we're gonna make a breakthrough for personalized therapies for pancreas cancer, it's gotta come from targeting these particular variants. And already, when you look at KRAS G12C, there are some very promising signs that we can start to figure out a way to turn off the light switch. So these are 3 different trials, 3 different drugs, and to target KRAS G12C. And adagrasib, about 1/3 of patients had shrinkage, 42% for this one, 21% for this one. Now, the responses are not durable, but remember, these are patients who have totally run out of options, that have limited life expectancy. So not only are we able to shrink these cancers and improve survival, but we're doing so with almost no side effects in most cases. So this is a major breakthrough, because remember, these patients in this setting have been heavily treated with very aggressive chemotherapy. So to be able to control their cancer, even for a short period of time, with minimal side effects is a big breakthrough. And I copied this out of our national guidelines. This is our national NCCN guidelines that we as doctors follow. When you look at useful in certain circumstances, already these KRAS inhibitors are making it into our national guidelines. So, I think this is telling us that, at least for that rare variant, we now have therapies available to us. Could we do better? Yes, we could. We'd love to see people having more durable responses, but that's a great place to start. Now, if we're going to make breakthroughs, though, we've got to get to the big three, G12D, V, and R. And here's what's happening in 2024. There are drugs coming into the clinic right now that are designed to attack these types of mutations. And they do so with lots of different strategies. One is what we call high potency targeting, where the drug can just flip that switch into the off position and override it. There's degraders, where you can basically degrade that light switch so it no longer exists and shuts the light off. And then we have these things called molecular glues and covalent bonds with complexes. All this is a lot of drug chemistry lingo to say basically they're coming up with more and more creative ways to figure out ways to turn off the light switch. And so that's exciting. And I just made this list of all the KRAS inhibitors coming into the clinic this year. Many of them are already in the clinic. Some I have not listed here. But this is all to tell you that these studies are ongoing and patients are now getting on trials of these KRAS inhibitors, and we hope to learn a lot more over the next year about how well they work. But I'm cautiously optimistic that this will be a major paradigm shift. So I did promise I would talk about those rare patients who do not have a KRAS mutation. We call it KRAS wild type. What do we do for them? Well, there's good news for these patients as well. And that is, these patients tend to have other mutations that are actionable with other drugs. So across this column, you can see different types of mutations or alterations that are present in these wild type tumors. And then all these drugs that are now entering the clinic are already in the clinic where we see responses. So we won't focus on the details of these drugs. The key thing I know is if the tumor is wild type, look for some of these other rare alterations. And in fact, when you go back to our national guidelines, they even say now genetic testing for inherited mutations is recommended, but do molecular profiling. This is our national guidelines now. So every pancreas cancer patient should ask their doctor, have you done the molecular profiling on my cancer? Can I see the report? And carry that with you, because that's something that typically lives outside the medical chart. So it's important for a patient to have that printed out and keep that on them, because oftentimes when patients go to get second opinions, that's one piece of paperwork that doesn't make it because it doesn't come out of the chart. So just to wrap up, I think precision cancer medicine for pancreas cancer is finally becoming a real thing, entering the clinic. I strongly recommend, and consistent with our national guidelines, genetic testing of the patient. We call it germline testing, to look for inherited predisposition for pancreas cancer. I think this is an important new addition to our guidelines. It is also can make the patient potentially eligible for certain types of therapies. And then I do recommend molecular testing as well, just like I talked about. I'm not going to focus on our actionable targets, but this is a list of all the things we have in 2024 where we have therapies available for these patients. And I'm particularly optimistic about our new approaches to target KRAS, which could open up a whole range of new therapies for patients with pancreas cancer outside of our traditional cytotoxic chemotherapy, which nobody likes, not even the guy who gives the chemotherapy, who's me. So with that, I will wrap up.
Okay, thanks very much. Do we have any questions? I've got a couple. I noticed you didn't talk too much about cancer vaccines, which seems to be kind of the latest, biggest thing right now. Do you have a view on that?
I only didn't talk about them because you gave me 10 to 15 minutes. So I exhausted my time talking about this. But yes, there is some early data in the realm of cancer vaccines, indicating that you may be able to use cancer vaccines to generate an anti-tumor immune response. And there are some hints from some early pilot studies that could potentially lead to beneficial impacts for patients. But these vaccine trials are in early days. I'd say we're probably a year or two away from making them widely available. I also think that when you talk about vaccine trials, I think the field has been held back because these trials tend to be conducted in patients who have more advanced disease. And it may be the case that vaccine trials are at their best where the patient has minimal disease, right? And so I think those types of trials are a lot more difficult to run. And because patients have maybe have just had surgery, it would take sometimes years to even show that you've altered outcomes for them, right? Compared to, say, a control. So it's early days. I'm cautiously optimistic, but there are some signs that maybe we're making progress in this way.
Okay. Yeah, just by way of background, I'm a pancreatic cancer patient, and I am in a clinical research program for a cancer vaccine, a neoantigen peptide.
Yep.
And I just had my 6th dose yesterday. I had 5 doses with no side effects, and I had the 6th one yesterday, and it just wiped me out.
Yeah.
So I don't know what that means. I hope it's good. Hope it means that that my body's reacting to it. But there's nothing on my scans at this point. I'm kind of an early in the process, so hopefully I get a better result. Alan Morris has a question.
Oh, he's on mute though. And Roger, I wish you well with that vaccine trial. I don't know which one you're on, but we did see some publications late last year, particularly coming out of that multi-institutional team, and then Sloan Kettering has one as well. And the data is intriguing, but I think it's tough to run those trials. There's a reason why it's been super slow to the clinic.
Yeah, mine is through the Jamie Leandro Foundation in San Francisco.
Uh-huh.
And it was difficult to get into that process,
Yeah.
but they seem to have had pretty good results. But like you say, it's so early, who knows? Ellen, if you want to unmute, you can ask your question.
Hi, Dr. Strickler. How are you?
Well.
Good. I see you're part of the molecular tumor board at Duke. I have questions about that, but I don't want to start with that. I'm going to make some statements. If they're wrong, tell me. Pancreatic cancer is viewed as a cold tumor. Is that correct?
Yes.
Okay. And is it also correct that the conventional wisdom is that the cold tumor in the cold versus hot dichotomy is the worst tumor to try to evoke a vaccine response? Is that correct?
Yes, that's true. That's why.
Okay.
Yeah.
Thank you for giving those 2 axioms to me. You are aware, and everybody's aware, that the only therapeutic vaccine that has been successful to date has been in prostate cancer. Needless to say, this audience this site started as a prostate cancer site, I think, but Brad can correct me if I'm wrong. Prostate cancer also, correct me if I'm wrong, is considered a cold tumor. Is that correct?
Yes.
Okay, and you are, I'm going to ask the question again. You're aware that the only therapeutic cancer vaccine to date is in prostate cancer, right?
Yeah, I mean, there have been promising signals in other tumor types, particularly melanoma.
Okay. But that's with ICI. That is when it's combined with ICI. So it's not a pure vaccine kind of situation. Okay. So that is a paradox. Would you admit that that's a paradox, that everybody thinks that a cold tumor is the one that is least likely to get a therapeutic vaccine for, and everybody's chasing? In fact, Moderna and BioNTech have been chasing melanoma for 30 years unsuccessfully. Okay, now that I started with that, I'm going to a punchline. It turns out you weren't present, but there is I believe he's an interventional radiologist, but people can correct me if I'm wrong. There's a guy named Dr. Gary Onig, and he is using cryotherapy to produce an auto vaccination. In other words, for example, with prostate cancer, he stuck a needle into a bone, just a 1 millimeter focus of bone prostate cancer. And he, believe it or not, evoked a cure in himself. And what do I mean by cure? He has no evidence of disease times 5 years, yet he was a stage 4 metastatic cancer. Now, the reason I'm bringing this up is I'm segging it with pancreatic cancer, which is also a cold tumor, of course,
Right.
with a much worse prognosis. Interestingly enough, just as one aside, I'm sorry for this tangent. One of the hallmarks of pancreatic cancer, correct me if I'm wrong, is desmoplasia, the actual fibrotic thing, right? It turns out, unbelievably, prostate cancer has a uniqueness as far as that goes too. It is the only cancer known to man, solid cancer known to man. Are you a general medical oncologist?
No, GI medical oncologist.
Oh, GI. Okay, so you don't know this. It's the only cancer that goes from stage 1 to stage 2 by virtue of a positive digital rectal exam. That's my tangent. I'm sorry for inserting that. It's one of my bugbears. But let's go back to Gary Onig. Believe it or not, he did the same thing for pancreatic cancer. And unbelievably, and I'm gonna ask questions about this. Unbelievably, this person had liver mets, and they had multiple liver mets. And he had a complete response, complete resolution of all his liver mets with his cryotherapy, whatever. I guess I'm going to stop there and ask you a question. Have you ever seen, with any vaccine tested to date, a complete response of metastases? Have you ever seen that?
No, I've not seen that, certainly in the GI world.
Okay, excellent. You need to see Gary Onig's stuff, because it happened. And it happened with an unsophisticated, like, for example, Roger is doing some neoepitope thing, which I'd love to drill down on. And Roger, on another date, if we could have a conversation, I want to know what your neoepitope formula is. But not knowing any kind of epitope formula for a vaccine, just using the lysate of a person's own pancreatic cancer, he was able to induce a complete response. Since you're a researcher looking at trying to get at a cure of pancreatic cancer, you need to talk to Gary Onig, at least in my opinion, because he was able to do that. I think there's a question in there somewhere, but I'm not sure. What's my question? My question is, come on, this speaks to the fact that cold tumor is the best tumor to go after a vaccine for. And I know why, but you won't ask me why, and nobody will.
All right. So any comment on that, John?
I think in order to make progress in the field, we need to learn from these N of 1 cases, right? N of 1 cases are what inspire research. They inspire, funding for these kinds of studies. But that we can't move the needle. We can't change the world with case reports. We have to hypothesize that this intervention will, lead to a certain outcome, and then we need to get funding from it, ideally from a foundation source, because this is obviously a type of intervention that doesn't have a reliable funding source, right? So you'd have to go through PanCan or ANCAN, whatever it may be, and we need to bring larger pilots in, just like some of the exciting tumor vaccine results that we saw last fall were published off of a 20 patient series, right? Because once you show that positive effect, then you can take it a step further and make it standard of care. So you don't have to know somebody to get it. You can get it as part of our national guidelines. So, this is all part of the process, and we can learn a lot from people who are willing to, stray from outside of the norm or convention, because with pancreas cancer, the convention is just not acceptable, as I'm sure Roger will tell you. It's the long-term outcomes with standard of care with pancreas cancer are unacceptably poor, and we need paradigm shifts. in order to really move the needle with this very difficult to treat disease.
On that point, you had mentioned the mutation testing of the tumor. And I know PanCan has a Know Your Tumor program, and I was in it. We went through Tempest, and then I went through Boston Gene. But since I joined Cancer Patient Lab, I learned about proteomics, which I didn't know anything about. No one had ever told me. And I got a proteomics test, and it found a whole bunch of new therapies and targets that were useful for me. Do you think that's something that might become part of standard of care anytime soon? Because I think it was immensely valuable to me.
Yeah, I mean, we're going to run out of, when you run a DNA-based test, you're talking 300 to 500 targets. Some of them are actionable, some of them are relevant, some of them are not. But, we're going to run out of targets and therapies eventually. And then you get to RNA-based tests, and which you have through Tempest and some other assays. And there you can get into expression profiling and whether those could be used to predict sensitivity or resistance to certain types of therapies. But you're right, proteomics gets to a whole nother level. And I think that the bioinformatics and the machine learning and the AI you need to truly be able to take that information and apply it to therapy. Like, we've only now just developed the computing capabilities to understand even how to use that very complicated data to guide therapies. And I suspect to really capture the value of it, we're going to need a whole next generation of therapies, right, that are designed to target those proteomic signals. So yeah, I agree. I think if you're looking at where we're going to be 10 years from now, I would not be surprised if you're starting to see these proteomic readouts driving therapeutic decision-making, particularly for difficult diseases like pancreas cancer.
Thank you. Brad has a question, it looks like.
Yeah, just staying on this topic of more diagnostics gives you more data, it gives you more treatment options and more personalization. What about liquid biopsies? I know your colleague at Duke talked to us about some AI he used to come up with a kind of a multi-dimension biomarker that was based off of a blood biopsy. And so to the extent, and we also recently had a discussion with Tony Magliocco, who mentioned that there's now RNA liquid biopsies, and that provided visibility into a whole range of things where you might not have enough to look at DNA, enough circulating tumor DNA to look at it. But with RNA, all of a sudden a liquid biopsy becomes much more viable because there's a lot more that you can read. And that gets you closer to almost like real-time monitoring. That, blood biopsies we can take quite frequently, whereas tissue may be hard to access or what have you. So what are the advances that you're seeing that might be available now or in the next 6 months, the next year, that would be in that domain of liquid biopsies that would allow tighter, closer monitoring so that you can sort of observe what's happening and then maybe nudge things and approaching a real-time basis?
Yeah, I mean, I think I love this question because circulating tumor DNA has also been a major new diagnostic technology that's entered the clinic in the last 10 years. And it's got its strengths and limitations. So the major strength is it's convenient. You just draw 2 tubes of blood. Unlike a biopsy, which many of you know what that feels like to get a biopsy, it's not particularly pleasant. You can sometimes get the readouts much quicker. It's easier to put people on a trial when you're using that liquid biopsy because the speed is so much quicker as well. And in general, the results are, for the most part, trustworthy. If you see a KRAS mutation in blood, you know it's real. It's not some bogus lab. So I think for that reason, it's got major advantages. However, when it comes to pancreas cancer itself, there are some major limitations for blood-based profiling. So as Alan pointed out, it's a desmoplastic tumor. So when you look at a lesion, say a liver lesion on a scan, it's 90% stroma. There's very little of that. It's actually active tumor content. So as a result, you think about that cancer, it's almost surrounded by a rubber ball, right? So it doesn't produce a lot of DNA into the bloodstream that can be captured on one of these tests. So the yield of circulating tumor DNA for pancreas cancer specifically is much lower than, say, colorectal cancer, which is another disease that I treat, which means that the test is just less reliable for identifying the targets of interest. So that's been a frustration we've had about bringing these tests in for pancreas cancer, and why I've tended not to use some of the commercial assays, just because I will order the test and it'll show nothing in the blood, but I will order the same test on tissue and it'll show me all these targets in tissue. That said, we are now entering an era where these blood tests are doing more and more. One intriguing application of the blood test is a hypersensitive early detection tool for pancreas cancer, right? If we're going to move the needle for pancreas cancer, we've got to detect this earlier, because it is very difficult to cure the disease once it becomes a bulky tumor. Once it's spread to the lymph nodes, it becomes a very concerning development for the patient. So if you can pick this up earlier, say 5 years before it became a full-blown cancer, you could potentially move the needle and protect people from a pretty aggressive malignancy. So I think where I see the greatest opportunity in the future with these blood tests is not so much the molecular profiling, but more on the preventative medicine, the early detection. Those kinds of tools could be a really interesting application for that type of test.
I have a question. I'm wondering where this fits in, exploring the off-label use of existing medications that, research has never looked at, for pancreatic or prostate cancer, but sometimes by accident and sometimes maybe just beginning right now through some artificial intelligence review or finding possible applications.
Yeah, so I mean, one of the frustrating things that we've got is that it costs a lot of money to develop a drug, sometimes billions of dollars. And if the drug is off patent, then that company can't recoup the cost of their investment. And so I do think there's probably some therapies out there, discarded therapies, natural therapies, where we just don't, we just can't get that type of research going because there's just not enough funding for it. And so that can be frustrating, because I think that not every new therapy has to cost $3 billion to develop, right? It could be something that's widely available. But it's a challenge because patients are obviously seeking out these kinds of therapies, and they're smart. They understand that there's never going to be a randomized trial. And sometimes there's what I call internet lore around certain things that aren't proven yet. And it can be very confusing to navigate that as a patient, because you don't know whether that product that you're researching is really, would be FDA approved if it had the financial backing, or if it's just as effective as sugar water. And that's a challenge I've got as an oncologist as well, because I don't have access to that data either. And I do hope that in the future, we will have better ways to fund research on therapies that don't have so much of a financial incentive, that don't have to cost $50,000 per dose, right? Like we should be able to do research on things that cost $5 a dose.
Rick Davis has a question.
Hi, Dr. Strickler. ANCAN has an interest in pancreatic cancer. We run a peer support group, which is a partnership with Canopy Cancer and Hirschberger, a couple of others. So it's peer-led. So if you have any patients, don't hesitate to send them over. We're running once a month right now, but we're about to move it to twice a month. And so I've learned a little bit about pancreatic cancer. I have two questions, one about BRCA and one about TMB.
Mm-hmm.
And BRCA's really should be interesting to the prostate cancer folks, because it's relatively common in prostate cancer, probably more common than in pancreatic cancer, although that's one of my questions, because you sort of skipped over the BRCA stuff in your presentation. So I'm real interested to know how often you see it. You also talked about germline BRCA in your slide, but not somatic BRCA. And my understanding has always been, doesn't really matter whether it's germline or somatic, it's still going to be sensitive to treatment from PARP inhibitors. So first question about BRCA is, can you talk a little more about it? And one big question for our prostate cancer folks is, what monitoring do they do if they've got if they're BRCA positive, germline BRCA positive, not in the tumor, but if they're germline BRCA positive, how do you monitor for pancreatic cancer? Like you can do, you can squeeze your breast to see if you've got breast cancer, and you can check for melanoma, you can do other things. What do you do for pancreatic cancer?
I love this question. They shouldn't have told me I only had 10 to 15 minutes. I have slides on this too. I had to cut out the immunotherapy section and the BRCA section just to stay in my time. But look, we do have FDA-approved therapies, or therapy, I should say, for germline BRCA pancreas cancer. It's olaparib, which I'm sure the prostate cancer folks on the call know that drug well. And that came to us out of a trial that took patients with metastatic pancreas cancer and randomized them to olaparib by itself or just standard maintenance treatment. And it was interesting because now all the patients had to have germline alterations. We'll go back to the somatic later. But what they found is that the patients who got the olaparib were put into a longer remission after completing chemotherapy than those people who didn't. One of the challenges with that trial, though, is it did not show a survival difference long-term for giving olaparib. So the drug was FDA approved, but it was because it delayed relapse of the cancer, but it didn't improve survival. So it's somewhat controversial. If it doesn't improve survival, should we give it? I think our feeling is that patients still benefit because they're spending more time off of chemotherapy. But that's why it's a little bit of a thorny topic for us around that. Now, just for the sake of the audience, germline means it's something you're born with. Germline BRCA mutations are basically a genetic predisposition to different types of cancer, including prostate cancer. Somatic means you're not born with it, the cancer acquired it in some way, and it's part of the cancer, but not part of the person. So it appears that those PARP inhibitors are truly only active in pancreas cancer patients who were born with that BRCA mutation, or the other one is PALB2, not somatic. Now, I understand that there's some literature around this in prostate cancer as well. I think probably I'm way out of my lane with prostate cancer, but my recollection is that those therapies, those PARP inhibitors, are most active in patients with germline alterations, particularly BRCA 1, 2, and PALB2. But you all can correct me if I'm wrong, since I don't treat.
Well, BRCA 1, BRCA 2, maybe ATM,
Yeah,
and maybe PALB. The conversation around PALB that we've had has been largely with Pamela Munster, who's really a BRCA expert.
right.
And she says, well, if it worked for BRCA, it's got to work for PALB too, but we don't really have the evidence. But we know for prostate cancer that it doesn't matter if you're germline or somatic.
Yeah.
And so it's really interesting for me to hear that it may matter in pancreatic cancer. So if you found a somatic mutation, if you found a mutation in say from Tempus Foundation Medicine BRCA, you'd have to go check if it was germline before you use
That's
a
true.
PARP inhibitor. Whereas in prostate cancer, you wouldn't
Exactly.
do that. You wouldn't need to do it.
Although I would say that in prostate cancer, you would probably still want to test whether that was
Well, yes.
well. But But it wouldn't,
Prophylactically for relatives and what have you, absolutely agreed. But not from the standpoint of whether it would work or not.
right. So, yeah, I mean, I think that's why we're recommending that patients with pancreas cancer get germline testing. Hopefully Rogers had his done, and I don't know which vendor you used for your test, but you know, it's something that's increasingly standard. It's seen in about up to maybe at most 5% of patients have a germline BRCA1, 2, or PALB2 mutation in pancreas cancer.
Yeah, I don't. Say, I want to shift gears a little bit. Just in my case, I caught my cancer at stage 2. It was resectable, had 6 months of chemo. I'm NED right now. But after I found it through a Gallery blood test.
That's interesting.
And I had to fight to get it. I'll just tell you that 2 years ago, at least, doctors were not recommending it, and some of them were just hostile to it. But that's what found it, and it was pretty significant. And in fact, I like to tell people the story about this radiologist that I contacted. He called me up one day, and I assumed he called me to schedule some sort of radiation. He said, "Oh no," he said, "you're not a candidate for radiation." He said, "I just had to talk to you because you're the luckiest person I've ever met." Because it was distal tail. He said, "You never would have known about this otherwise."
Yeah.
So I guess that leads me up to early detection, because that seems to me to be, one of the best ways to fight this disease is just early detection. What do you think?
Well, that was my point with the blood-based testing before. If we're going to move the needle I mean, pancreas cancer is extraordinarily difficult to cure. And, I'm thankful that they found yours early, that it was distal tail, because that's a lot easier surgery than some of the other surgeries we offer patients in your cohort, right? But that's going to be the way we can do better with this disease. We have to catch it earlier and earlier. And, you're right. When you talk about these early detection assays, from my perspective as a practicing medical oncologist, I've only seen patients who've shown up in my clinic who found their cancers through these tests, right? I've never seen a false positive, because you can't get to me unless you have a true positive. I think one of the challenges that I was talking to a primary care colleague, and I think the challenge they've got is that when you're dealing with a low prevalence tumor, you need a very high sensitivity and specificity test to avoid a lot of false positives. And I think the primary care doctors are worried that if the test is not specific enough, we're going to be terrifying a lot of people and they don't actually have cancer, or they may end up getting a lot of extra biopsies and other things that they wouldn't otherwise need. And so I think that's got to be something, that's where the pushback is coming. It's actually understandable when you think about the fact that, like, the numbers have to be really tight on one of these tests to avoid a lot of unhelpful follow-up studies and biopsies. But from my perspective, your story is the story I've seen, which is that these tests have been used to find things earlier than people would have otherwise found them.
Yeah. So Brian, I see you have a question. Are you able to ask it, or are you otherwise indisposed? If so, I'll read it.
Can you hear me? Can you hear me?
Yeah. Hey, Brian.
Yeah. So I was recently at a symposium. Sorry, I'm on my phone. I can't do anything about it. Anyway, I was at a symposium, and this doctor was talking about radioligands in pancreatic cancer. And of course, this is a field that's been pursued for quite a while in prostate cancer. So curious to get your thoughts on the use of radioligands in pancreatic cancer.
Yeah, well, I mean, radioligands have really gotten a big boost from prostate cancer work, right? Where we do see the radioligands have been successful for that tumor type. In order for a radioligand to work, you need a target that's universally expressed, that's not on normal tissue or low expression on normal tissue, and then an effective therapy tagged to it, right? So I do think radioligands, there's no reason why you could not develop a radioligand for pancreas cancer, right? You know, whether it's CA19-9 or some other protein, you just have to find the protein and the target and engineer that molecule. I'm not aware of anything being developed, though, strangely enough, for pancreas cancer, at least in this radioligand category. So maybe there are some technical issues that make that drug development a little trickier, but to me, it is fair game as a potential avenue for novel therapies.
Telix is actually working on something for Pancan, but it's tied in. I believe it's tied into what they're doing for neuroendocrine with dotatate. So it wouldn't be applicable, obviously, to the exocrine Pancan folk, but it might be applicable to the neuroendocrine Pancan folk. I know this because I have an old school friend who's a professor and a pediatrician in Australia with Pancan, and I just reached out to Telix on his behalf and connected them. So it's not here yet, but if you want to know more, then I'm happy to connect you to Telix.
Well, you're absolutely right. We do have radioligands. We've got lutetium dotatate. Lutathera is the trade name for it. That is available to us. I believe it is FDA approved, and that we use routinely. But there are new radioligands that are coming in the clinic specifically for the neuroendocrine type. But what I was referring to was specific to the exocrine pancreatic adenocarcinomas.
Yeah, and the work that Misha Beltran's been doing and Rahul Agarwal on DLL3, they've also used that, I think, in some pancreatic neuroendocrine patients as well. What percentage is neuroendocrine and what percentage is exocrine?
I think 90% would be my guess is exocrine pancreas, and in this adenocarcinoma category. The neuroendocrine subtype is pretty rare, and they have a much different natural history. In general, do much better with therapies. Tends to be fairly chemotherapy sensitive, and then we of course have these radioligands available. So if you would never wish a pancreatic malignancy on your worst enemy, but if you were to choose between the two as a patient, you would rather have a neuroendocrine type, I would think, than an adenocarcinoma. The adenocarcinomas also just tend to be far more symptomatic. I'm personally convinced that there is a cytokine pattern that just makes people feel awful with adenocarcinomas. It affects people not just physically, but mentally and emotionally. And I'm sure in your NCAN work, you've just seen how it can affect not just you physically, but emotionally, mentally as well.
Oh, yeah.
And I think cancer is much bigger than just a physical phenomenon. The body knows something's wrong and it feels that inflammation, and the mind, the mind absorbs that with, in its own way.
I'm really glad to hear you say that because I went for probably a year of having that feeling, and everybody is like, you're such a hypochondriac, there's nothing wrong with you. And I just knew something was off. Debbie has a question, it looks like.
I do. Thank you, Roger. Hi, Dr. Strickler. I am a huge fan of targeted therapies. I have been on imatinib for coming up to 19 years, and I'm still on first-line therapy. Realistically, what is the sort of outlook in terms of the KRAS inhibitors that are in pipeline that are coming to market? So I see there's one that's sort of pan-KRAS. I think that Boehringer's bringing that out. Is this going to be a kind of a game changer? I mean, there's been targeted therapies. A lot of these are already out in lung cancer, and I've worked on quite a few of them over the years. Yeah. So
yeah, I mean, I think the imatinib story is the poster child story
it is
for how amazing and life-changing targeted therapy can be.
Absolutely.
And that's, of course, for CML, and which is a type of leukemia that's very clonal, very dependent on that target.
Yes.
The problem with solid tumors is that they tend to have a lot more mutations in them, and they can morph, they can be chameleons, they can change under that selective pressure of therapy much more quickly than, say, CML can under the treatment of imatinib. So as a result, we see the outgrowth, we see the evolution of resistance much quicker in most solid tumors than we do over in, say, CML treated with imatinib. And that creates a challenge for us because we are always chasing that evolution. Now, in the case of lung cancer, we have therapies that will hold the disease back for years. And then every cancer has its own kind of, for lack of a better term, its own natural history, its ability to evade that targeted attack. Interestingly, when you look at that KRAS G12C story, which I showed you a little bit, lung cancer was most sensitive to it, and the least sensitive to it was colon cancer, meaning colon cancer could rapidly morph into a resistant type of cell, and pancreas cancer was somewhere in between. And I think in order for us to make long-term, like to truly turn pancreas cancer into a chronic disease that you just take a pill every day for, would require us getting smarter about what that pattern of resistance looks like and being able to short circuit it and prevent it in a way, because we're a long way from the imatinib story with these targeted therapies in solid tumors. But what I really find promising is that, you know, we are making progress here. As I showed you in 2013, we had nothing. We didn't even have PARP inhibitors at that point, right, for the BRCA germline tumors. We are now bringing in therapies that are beginning that attack, but now the next level, 2 years from now, once those therapies kind of establish their worth, then we're going to say, okay, do we pair this thing up with something else to make it work better? Do we give something else to prevent that outgrowth of resistance? What does resistance look like? But this is where we're headed. Right now, we're just having any treatment at all is a breakthrough for pancreas cancer, which is kind of sad, but also encouraging at the same time.
Yeah, that's how lung was when I first started working in the space. I work with life sciences, so in the development and commercialization of these molecules,
Right.
I saw the high unmet need in lung cancer initially, which has now become a space where there are many, many treatment options, even for the very super rare, once in a career, people like intract fusion cancers. What I see now is almost a situation where the oncologists can't keep up with that treatment landscape, and the patients actually have to become empowered to know their own disease, which is important as well. Okay.
You're right. Patients need to be empowered, and it's okay to get a second opinion, and make sure that, you know, things that they're hearing and seeing and understanding that those are calibrated, right? Because you're right. I don't know how a doctor that treats every single cancer under the sun, CML, breast cancer, you know, multiple myeloma, pancreas cancer, all the different subtypes of lung cancer, you know, to know what's coming out 2 years from now is practically impossible for somebody who has to treat literally hundreds, if not thousands, of different diseases in their clinic. So, and I personally get referrals, and I work with those doctors to say, hey, look out, in 6 months, there's going to be an FDA approval for drug X. And they're very appreciative to know, okay, this is how we're changing. This is what's on the horizon.
Okay, we got time for one more question, and I see Richard has his hand up.
Thank you. That was really a fascinating presentation. I guess just to follow up on the question first, are there any of the mid-stage to late-stage KRAS trials that you're excited about? And second, you know, after that question, if somebody escapes, is there any history or knowledge about if somebody escapes one of the KRAS inhibitors, of which there aren't many yet, do they hop into a different KRAS mutation, or do they sort of hop into unknown territory, with some other kind of mutation that's not in any of the target lists? But I guess the first question first is the BI question, or whatever.
I should have just presented all my slides because I had all that resistance data in there too. So yeah, I think one of the challenges of developing drugs for KRAS is that, as I said, most of the time when there's a mutation, it locks the switch in the on state. But the drugs we have currently in development, the ones that I showed you for KRAS G12C, lock the protein in the off state. Well, the protein has to be in the off state to be locked in the off state. And so that's probably why it's kind of struggled to be active in certain contexts. In order for us to build, I think, a next generation of therapies, we need to be able to attack a light switch when it's in its on position. And there are drugs now entering the clinic that do this. I'd point out the Revolution Medicine pipeline, which is designed to attack these so-called RAS-on variants. And already they've showed some interesting data in pancreas cancer. So I'm really excited about that particular technology from Revolution Medicines. But this is an era where there's going to be countless different approaches taken for that. Now, the second part of your question is, what does resistance look like? And there's two types of resistance. One is acquisition of new mutations, outgrowth of new mutations that rescue the tumor to keep the light switch on. So if the mutation is KRAS G12D, one way to escape would be to make a different RAS mutation that is not impacted by that drug. Like maybe it's KRAS G12R will outgrow. It'll be present in 1 in a million cells, and it will kind of, by process of selection, kind of grow out under that attack. The other interesting biology that we've learned is that there are receptors on tumor cells that can also rescue the disease by activating what we call wild type or non-mutated processes. So like when you inhibit KRAS, you can actually activate normal proteins in the cell to keep the cell turned on. And so that's another tricky piece of biology that we'll have to figure out for certain types of tumors. That's particularly a problem for colon cancer, a little less of a problem in pancreas cancer, but it can rescue tumor cells even when you think you've got your light switch in the off position, right? You can do that.
Well, like for Gleevec, there are, a whole host of downstream mutational drugs that deal with, whatever the c-KIT mutation or whatever the first set of mutations, it sort of hopscotches along with it. But, I guess if you get a constitutively active cell, you're in trouble. And if you get, an alternate pathway, you're in trouble. But if it's a KRAS mutation, is there any sort of insight about whether it goes to a different potentially druggable KRAS mutation, or there's just no history or not enough knowledge yet?
It will typically grow out as multiple simultaneous mutations. So you think about a cancer being in maybe 5 spots in the body. Each spot will have a different clone grow out that's resistant. So you can have outgrowth, and that's why it's so challenging with some of these GI cancers like pancreas cancer, is you can have multiple simultaneous resistance alterations. So even if you could target one of them, well, you'd have to target all five different ones simultaneously. It becomes very tricky. Now with
you might have a C and a D at the same time, for example.
A C and a D and a V and an R, plus a KRAS wild type amplification, plus a BRAF mutation. And you notice all those mutations are designed to rescue the same pathway that was activated by the original KRAS mutation. So they won't, they won't go through a different pathway. They'll activate the exact same pathway in that cancer.
If I could ask just sort of one clarification about the first part of your answer, which was, you said that these KRAS drugs, or at least some of them, typically target KRAS off. Is that like it's in an off conformation, and from time to time it gets into the off conformation, and that's the moment you target it?
Yes.
And then if it's on conformation, it's not targeting some part of the path, some part of the receptor that is always available. It's targeting the part that's only available when it's on, when
That's
it's
right.
off.
So the concept here is called cycling, like a switch turns off and a switch turns on. And certain types of mutations are like always in the on state, and certain will flip back and forth between on and off. So if you have an off inhibitor, a protein that is flipping over to off, that's when the drug will bind, but not when it's in the on. But there are now drugs being developed that can target it in the on state as well, which is huge.
Oh, interesting. That's
Yeah.
really interesting. Yeah, because KRAS is so flat, it's a hard molecule. I understand.
You're absolutely right. That's why it's taken us 40 years to figure out how to build drugs for it.
Wow. Thank you.
Okay, well, I want to thank you, Dr. Strickler, for being here. I'm going to hand this back to Brad.